habitat issues. We'll have two speakers. It'll be the same format as yesterday. After the speakers, we'll have time for discussion. Our first speaker is Dr. Takaman.
Thank you. My name is Yasuhiro Takemon, and I'd like to show you, one topic. Yes. The title is the Habitatology for Linking Riverbed Geomorphology and Ecological Functions. My colleagues are now engaging in the studies on the geomorphology and also the ecological functions. And as to the geomorphology, it is very closely related to the disasters and the water resources. So as to the sediment, we have to make a management in an integrated manner with the disaster prevention, water resources, and environmental requirements. And today, I focus on the ecological evaluation of channel geomorphology because this requirement is not always established enough. So in order to say what is the important for the ecological conservation, we have to show this geomorphology should be for this purpose. Such kind of interrelationships should be shown. And today's topics is how to make such kind of the methods in a proper manner. Most of you are already familiar with this kind of pictures. The ecological functions are always closely related to the physical habitat structure, not only within the channel, but also the bankside geomorphology and also the hyporheic zone properties. And the important thing is each portion is not always independent. They are all rated in dynamic structural manners. And from now on, I'd like to show you the habitat concept. Habitat structure is not always identical to the geomorphology. Because the habitat is always has the characteristics of the following manner. First one is concept of location. Location is like this. For example, within the river, the upper side and the tail side is very different for the inhabitants. And the river head and the river tail is the common word within my presentation. Now, from now on, I'll show you how they are different. And as to the river head, the water will penetrate into the riverbed, so-called downwelling zone. And on the contrary, the river tail is always related to the upwelling zone. And this is very important for the inhabitants. And the next one is the combination of the geomorphological features. Some inhabitants will need both geomorphology for completion of their life cycle or for the completion of the rural daily life. It depends on the species, but anyway, not only one component are important, but the combination also important. And the third one is the age. Even though the superficially the same geomorphology, but the meaning for the inhabitants are different. If the habitats continue to be for a long time, and from the creation to the vanishing, the history of the habitat should be should have meaning for the inhabitants. So the young and old concepts are very important. And so now I would like to make questions how to link among sediment dynamism, geomorphology, and habitat structure. In particular, in terms of the location, combination, and age, these concepts should be included in the target habitat structure in order to make a healthy river geomorphology. For this purpose, I proposed one terminology, habitatology. Because geomorphology is a bit different from the habitatology in terms of the characteristics of the habitat. The target parameter should be habitat features in case of the habitatology. It means this science is for analyzing habitat structure and its maintenance mechanisms. And this is the habitatology method. At first, we will make a classification of habitat structure. And the second stage, we will evaluate. And the third one, we should make clear the geomorphological and hydraulic mechanisms for creation of the habitat structure and maintenance. After that, we can apply the results to the actual works in the rivers. And this is the flowchart for completion of the habitatology. I always show my students to which part do you want to engage in. And this is the outline for the application of the fields in the practical sense. Yes. Excuse me. I think you are very familiar with these subjects. How much amount of the sediment load related to the riverbed geomorphology or flow regimes are more closely related to the targeting factors. But if we look at its ecological function, we need a concept of the habitat. And when we make a good ecological features, we should think from this way, from bottom to the upside. And in order to make the good management, we have to research these subjects. So ecological evaluation of the geomorphology and identification of the appropriate sediment load and flow regimes in order to create the appropriate riverbed. This is the today's menu of my presentation. I'd like to explain about the examples of the application of habitatology. First one is the habitat location and age at the reef head, because the reef head is very key character of the geomorphology for the oviposition sites of insects and spawning sites of the several kinds of fishes. Interestingly, they need exactly the same location. Of course, the longitudinally they are different, but as to the location within the reach, they use the common features for their reproduction. And another example for the mussels and the bitterlings. Bitterlings in a kind of like caprinid fish living in the lower stream of the rivers. And their habitats are always related to the terrace ponds. And the ponds has the age from the creation to the vanishment. And now I'm focusing on the age feature. The first one is the oviposition sites of the mayfly. Ephemera strigata is the target species. They will make reproduction in this part of the bar structure. This is upper side and the lower stream. And they will make a big swarm for making the oviposition here. And another example is the goby fish spawning red. This is male and this is female. And they will make a nest on the side of the stone and make this kind of egg mass. They will deposit egg mass on the side of the stones. However, the location of the stones always concentrated to this part. And this part is identical to the place of the mayfly oviposition sites. Always the leaf head. And in order to make clear the exact the hydraulic or the chemical properties of the leaf head, we made the permeability test using the Packer methods, and we can get the current speed of the interstitial water using the Darcy law. We need only the hydroconductivity using the difference of the gradient of the water and the interstitial K value. Then we can get the expected the current speed. And using this method, we measured along the bar and inside the bar. And then the hydraulic conductivity shows the high values only along the bar head and the leaf head areas. And blue circle shows the spawning red of the goby fish, and red circle shows the oviposition sites of the mayflies. And interestingly, they are gathering along the place of the high hydraulic conductivity. And as a result, the current speed of the pore water are very high at this portion. And this is related to the high DO concentration of the interstitial waters. It means the egg laying in this area can accept the high concentration of the dissolved oxygen, and as a result, the survival ratio of these animals will be very high. And as to the reproduction sites of this mayfly is restricted to reef portion. However, their nymphs will require pool and sandy bottom for growing. This means the reef and the pool sequences are very important for these insects. However, the reef head must be high permeability in the sediment feature. This is the conclusion. Mayflies and gobyfish need fresh gravel bar with high permeability. These features always corresponding to the bare land, bare sandy bar. If the sandy bar becomes old and vegetated, the permeability will decrease in amount, and the suitability as the habitats decrease. This means for these animals, the riverbed must be fresh. And the next question, how fresh is enough for them? This is the next question. And in order to answer this question, we have to make the monitoring of the geomorphology and make the correspondence between the time after creation to the suitability of the habitats. And this kind of works has done in the Tenryu River, using the material of the sweet fish, ayu fish. This fish is very famous in Japan because the people and the fishermen fond of this fish because of the taste is very sweet and not sweet, but it smells like the watermelon. That's the reason why this fish is so-called sweet fish. Taste is not sweet, but the smells like sweet. And they will make spawning at the reef head also. Reef head, like this egg. The diameter of the egg is only 1 millimeter. And this is the hypothesis why they need the very fresh the bar head. The bar head will be created during the large spades, and in this state, the erosion will be occur in the deep pool. However, after creation, if there is some small spades, the bar head portion will be eroded, and it means this part becomes very hard and armored by the small spades. In this case, this is not so good for fish spawning red because it is very difficult to dig out. At last, if this situation stayed for long period, during the low discharge, the small particle sediments will deposit and filtered in the this reef part, and the permeability becomes less. In this case, the DO concentration must be less. This is the hypothesis. And in order to test this hypothesis, we made the observation using this kind of the interval cameras settled on the tower and 6 meters high from the riverbed. And it is very difficult to climb up, so we hired the specialist climbers to settle the cameras on the tower. And the Tenryu River, a rather big river in Japan, like this feature. And you see the low discharge is only 100, but the high discharge increase more than 8,000. It means the very high changeable rivers. However, as to rivers, you see already vegetated because of the sediment shortage by the dam trapping. Upstream, there are a series of the big dams, and the sediment are now very, very short in downstreams. And then, this is the method for the monitoring. We will make pictures. This method transported from the Walters laboratory. The Trent University is the original one, and we made an imitation. But there is no cliff in Japan, so we used this kind of towers instead of the cliff in Italy. And in order to test the riverbed softness, we used this kind of apparatus. This is in Japan we call shino. The sword-like nail and steel nail, and we will push into the riverbed, and we measured the penetration length, and this is the method, and we made the monitoring before and after this kind of small spades. First one is the after 5 cubic meter. First one is the after 4,000 events, and next one is the 800, and this one is the 500, and this one is 500. And before and after of the spades, we measured the riverbed softness or the DO concentration, like that. This is the pictures taken by the interval camera before the spades and during the spades and after the spades. Then you can get some ideas of the difference of the geomorphology before and after. This small gravel bed vanished after this spades, and we can see this geomorphology must be very fresh. Just after the spades. But the others, like this part is not so different. It means this part is the very old age comparing with this one. And this is the results of the riverbed softness. We always measure the riverbed geomorphological features, environmental conditions, upper, middle, and lower. The riffle head, riffle middle, riffle tail, and always the conditions are different. In September, before the small space of the 800 cubic meters, the values of the riverbed softness is 6.7 centimeter on average penetration, 7.2 centimeter penetration, and in the riffle tail it is more softer than the upper, 10.6 centimeter penetration. And after the 800 cubic meter spades, the upper side, the riffle head becomes harder from 6.7 to 4.5 centimeter. It is very hard. However, on the contrary, the riffle tail becomes softer than before, 11.4 centimeter. This means as my hypothesis, during the 800 cubic meter spades, the stones on the upper side transported to the lower and accumulate in the lower side. That's why this part becomes softer. And after 500 cubic meter space, the older part becomes harder comparing with the before. This result shows the smaller space make the riffle harder, and for the spawning rate of the eyefish, they need the soft riverbed more than 10 centimeter penetration. It means that already this part becomes very bad for the eyefish. And as to the DO concentration, this is very interesting because some riffles already has the high DO concentration at the riffle head. Even though some sediments are already stacked, but at this part, the high maybe permeability is less, but the high water current are maintained because of the gradient or other factors. Anyway, for the purpose of the respiration, this part must be very good for selection. And okay. And this is the method for the making the potential map of the suitable spawning reds. These data are made from the 2 sets of the laser profiler data. And when we make the difference of the riverbed altitude, then we can get this kind of pictures. The red one is the depositional area, and the blue one is the eroded area. So if we make a map of this kind of the difference, then we can get ideas. Oh, this riffle is very new and the deposited place. This place is eroded, so it must be hard. This kind of potential map can be used for the purposes. And actually, this is we found the egg mass of the spawning red, and this part locates at this red area. It means we can use this kind of map for the finding out a good position of the spawning red. And this is the conclusion of the habitat age in Tenryu River. Same as our hypothesis. The good conditions must be made by the big event, but according to the data, it must be more than 4,000 cubic meters, and this space corresponding to the 2 or 3 year return period. As to the smaller space, it must be less than 1,000 cubic meters, and this is the very common space. During the 1 year, it experienced more than 2 or 3 times. This means that this is the situation of the Tenryu River at present. More than 20 years ago, this condition can be exist maybe more than 2,000 cubic meters or so because of the big amount of the sediment. Now it is deposited in the dam site. And then, unfortunately, the time is limited, and maybe 2 minutes or so. Yeah. So, this is the story of the riffle, and the next one must be the pond, but unfortunately, the pond story is a bit, I should skip. I should skip. Pond has also, on the other hand, the pond must be a bit older. For the bittering, the conditions before 1 year should be more important. And as to the mussels, the conditions of the pond 3 years ago are more important. Because for the growing, the mussel require 2 or 3 years, and it means the abundance or the richness of the mussels should be related to the conditions of the 3 years ago. It means the pond must be long in the age for the good conditions. Like this. This is the conceptual figure of the bittering and the mussels. The habitat suitability must be best during the succession of the pond. And it means the geomorphology must be sustained for suitable for the suitable period. So not always the fresh the better. So it is very different, and we have to combine both. The riverbed must be, this place is very fresh, this place is very old. This kind of the mosaic manner should be the best for the river environments. And from now on, I have the method for realizing this kind of situation using the integrated sediment management. And there are two kinds of the approaches, source control approaches and the local control approaches. And the regional sediment management in the basin scale are also related to the situations. And as to this, maybe for discussion. Okay. Yeah, thank you very much for your attention.
Thank you, Dr. Takaman. I forgot to mention that Dr. Takaman's chapter isn't uploaded yet, but it will be part of the book. Thank you. Our next speaker is Daniele Tonina from the University of Idaho.
Good afternoon, and thank you for John to convene this section, and for the local organizer of the committee, especially Daiso and John, for giving me the opportunity, to me and also my collaborator Alessandra Marzadi, that is a postdoc researcher with the University of Idaho, and Alberto Bellin, who is faculty at the University of Trento in Italy. So far we have been looking at the gravel bed rivers from outside, looking at what's happening to the water columns, how the surface water and the flow interact with the sediment, and how so this interaction shape the rivers. And not only the rivers, but also create this area of erosion and depositions, how that interaction also interact with the main flow, the ensample mean, and also with the turbulence. What I would like to provide to you now is a different view. Instead of looking at the river from the that moves above the sediment, to look what's happening to the sediment, because a part of the stream water is actually flowing into the stream bed material, below and beside the rivers. So what I would like to show to you is that river morphology not only impact the characteristics of the flow in the surface, but also characterize the hydraulics that happen into the sediment. Here is the still picture that was taken by John Buffington and myself a few years ago at the St. Anthony Falls. And what we did, we had a transparent flume. This one? Okay. Excellent. We put some dye right here, and because of the high pressure zone that is always present upstreams, an object that is in a flowing motion, in a flowing fluid. We do have a high pressure that pumped the dye into the sediment, then the move an arc path, and then right downstreams it is pump the water out of it. But let's look at a movie where we try at the Center for Hydraulic Research in Boise. Let's see if we can go over there. So let's stop it for a bit. So we added dye in a different point over the crest of this dune. Okay. So what I would like you to recognize is the water is moving from the right to the left. Each of these dots then will have its own flow path going through the sediment. And there are two things that are very important. One is the flow that tell us the amount of reactant or amount of water that can enter the sediment, and then the residence times, how long that dye will reside into the sediment. These two quantities tell us how much reactant or how much water enter the sediment, and the second information, the residence time, tell us how long that parcel of water will be in the environment that is in the sediment. So a certain type of biogeochemical reaction may happen, but also exchange of energy, because heat that move from the river that is warmed up daily by the sun then will provide energy going into the sediment. And so the exchange that happens between the river and the sediment is very important because it characterizes the habitat below it. Notice that then we do have this very long flow path that we injected into the sediment. It will take quite some time to actually exit in the sediment. So depending on where we are, we have a different view of the system. Then let's look at another movie over here. This one is a little bit faster and should. Here it is. Sorry about that. We injected over here. It's very fast. Before we have the residence times that was in the order of 24, 40 hours. Now we have a very fast one that was on the order of only 4 or 5 hours. So that's also another important concept. The residence times into the stream bed are very, very long. So they are not the minute or few hours that we can have into the reach. So we have this idea of a scaling, a time scaling now between the process that happen in the surface water and the one that happen into the stream bed are quite different. So we have 3 times, or at least we categorize 3 types of hyporheic flow. One is the one that we just saw that are generated by fluvial bed forms, and it's called the fluvial hyporheic flow. Then we have the parafluvial hyporheic flow that is typically seen into below dry bars. So there is a head difference between upstream and downstream the bar. And then we have the floodplain hyporheic dawn that happen usually between meanders or through paleochannels that connect the different parts of the river. But what is hyporheic flow and what is hyporheic zone? So what is the way that we can characterize and measure it in the field? There were 3 methods. The oldest of all is the biological method, and it was because the biologists recognized at the beginning of the 19th century that there is a different type of organism that live near the stream bed, where the characteristic of the water into the pore is very close to the one of the streams compared to other microorganisms that lives, microinvertebrate that lives into the groundwater. So absence of presence of those organisms was an indication if that area around the river is hyporheic or not. Most recently, we start to use a geochemical method, and that is injecting dye, like salt or fluorescent, into the surface water, and then through wells or piethometers, we can pump the water out, do a chemical analysis, and if the concentration of that tracer is larger than 10%, it's considered hyporheic zone, otherwise it's considered the groundwater. Very recently, we started to use the so-called hydraulic method, and we use basically head differences. So we have potential head field from which we can generate the flow path. And the idea is that the hyporheic flow is characterized by the envelope surface that contain all of the flow lines of in-stream water that enter the streams, and then somewhere downstreams of laterally, those flow line return into the streams. very recently, Mike Gusev started to think, well, sooner or later, some flow path will always go back to the rivers, since it's the catchment of everything. So we should start to recognize that probably we could say hyporheic zone of 24 hours or an hyporheic zone of 1 or 2 days. So depending on how we want to study, one method is better than the other one. So it's not that there is one that fits all our applications. With that definitions, there is also how the zone can expand and how deep the hyporheic flow can go. So there are some micro scale hyporheic flow generated by like a boulder or cluster boulder, like the slide of yesterday of Thanos, where it shows us the formation of a turbulence eddy right after a spherical object that draws water from the streambed. That's the same characteristics that happen along a boulder or a log into the streams, but also biotic processes like the formation of reds into the streams. Those create very superficial and short flow line. Then we have the channel unit hyporheic flow that is generated usually by bed forms like dunes or pool riffles. And then we have the channel unit one, that this is usually due by the fact that the pool riffle and the dunes are in Legoral. So we have some that are larger, some that are smaller, such that some flow line will circumvent the smaller one, joining the river somewhere downstream. So we have a very long flow path. And then the larger one at the valley scale, that could be dominated by meandering or changes in the hydraulic permeability due to location of bedrock, and so changing of the thickness of the alluvial layers or changing in constraints into the river. And then below everything there is the regional groundwater. What I would like to do now is to go very quickly and see which mechanism generate hyporheic flow. One of the easiest ways that we can do that is to consider control volume that we set right at the stream bed, and then we have the two lateral sides that are in line with the flow direction, such that we don't have a lateral exchange. We do have the bottom part that is adhered to the bedrock or the bottom of the alluvium layers, such that we have water entering the control volume from upstreams, downstreams, and then the exchange with the river. We can do this bookkeeping using this water balance equation over here. Then if we suppose that the system is a steady state, then we can simplify everything as the exchange is equal to the derivative of the discharge into the volume per unit length. If we then use the Darcy's equation to characterize the flow moving through the streams, we can now derive each point as a function of dl, such that we have now that E is a function of three properties. And those three properties are, one is the change of the energy head on the system. This energy head contains the piezometric head, so is the energy above datum plus the potential head, and then we have the dynamic head. Depending on which system we are, the dynamic head and the piezometric head may be dominate the total head. In the case of gravel bed rivers, we found out that the piezometric head is the one that dominates, such that the water surface elevation could be a very good indication of the energy head driving hyporheic flow. Instead, in dune systems where the dynamic head, due to the detachment of the flow in the lee side, that dominate the head balance, and so the dynamic head start to have the most important functions. The second term is the change on the thickness of the alluvium or the width of the valley, so the confinement, as that characterizes how much the area of our control volume change longitudinally. The third one is the hydraulic property of the sediment. So if we do have an heterogeneous system, so there are areas that can let the water flow more faster and slower, and so we can have more water flowing through the sediment. And because of that, and then we have areas where, because we have less water moving through the clay here, we pump out the systems. So beside these three mechanisms that works very well in explaining the exchange in a steady state condition. We have two that are non-steady state. One is due to sediment transport, because we have erosions, we have release of pore water into the rivers, and where we have deposition, we have entrainment of river water into the sediment. And so we start to have that exchange called by Elliott and Brooks as a turnover. Turbulence also can create very fast pressure fluctuations, and dependent on some coherent structure that can flow over the stream bed, we can have that perturbation moving faster and slower into the sediment. So all of these processes, plus the video that we saw, show us that there is these interactions. So there is surface water that move into the stream bed. So why is it important to study that? And that is for different reasons. The first of all, and where most of the study of hyporheic started, was because of a habitat concern. Salmonid and trout, they lay the eggs into the streambed sediment, and depending on the species, the size of the particle, the depth of the water, the velocity, they can change between 10 to 50 centimeters below the original streambed. And so knowing how much oxygen goes there and how much energy as the heat goes there is extremely important for the development of the embryos. The other part is that in headwater streams and medium-sized streams, we have that the stream bed is the area into the river where we have a very high amount of microorganisms and biodiversity between bioorganisms much larger than what we have in the water column, such that any type of biogeochemical reaction occurring into the stream is actually happening between the benthic zones and the hyporheic zones. So knowing how much reactant goes there and how long they stay there, that provides us a way to determine what type of biogeochemical reaction occurs. To do that, when we followed the analysis started by Elliott and Brooks, to study the flow into the streambed sediment as a Lagrangian systems. So we developed 3D models and 2D models to study how much flow exchange between the rivers with pool and reef morphology. And that's another very important point that I would like you to retain, is that depending on the morphology that we are working in, we can increase or decrease the amount of hyporheic that is exchanged. Once we have the flow field, then we can characterize how long a certain parcel of water stays into the sediment, and with that, then we can characterize with an advection diffusion equations and heat exchange, then how the temperature regime change and distribute into the sediment. In here we have a snapshot during midday where the downwelling area are in red, so we have very warm water, but as we move through the system, it gets cooler due to advection, dispersion, and conduction between the water and the sediment, the solid matrix, and also the timing that happens. So we have that waves of hot and cold. Same type of analysis we can do now to model, in this case, DIN is dissolving organic nitrogen, and see the transformations. How well this model works? It works pretty well, and I will show you in a little bit some application. But first of all, I wanted to go with you to show that we have analytical solution now, that we can calculate what are the mean residence time, the standard deviation, and the distribution of the residence times just from the hydromorphological information of the surface water. That means that the morphology of the system and the discharge into the system are key element in order to know what's happening underneath. Here is dunes that we studied with groundwater. In this case, it's a gain in reach. We have the groundwater that moves into the system. Here is we have a low pressure, and where the groundwater converges. Here instead is the same system where is a losing, so water that downwells into the sediment may be lost into the groundwater. Notice that even though the hyporheic cell looks exactly the same, they are shifted over one wavelength, and the wavelength is the length between one low point to another one point, or between the crest and the following crest. What we did is then be able to link now the mean hyporheic flow, both downwelling and upwelling, just with some morphological characteristic of the streams. Here we have the head, the energy head of drop generated by the flow that can be calculated with some analytical solution provided us by in the 50s. And then here we have the length of the dune and the mean slope of the streams. With these two information, then we can, and knowing if the system is losing or gaining, we can see that the hyporheic velocity can be nicely characterized by a power law. The other important information that I wanted to show here is the ratio between the hyporheic velocity and the mean velocity into the rivers. In most cases, is below 1%. So the hyporheic flow is very small velocity compared to what's happening to the streams. Here I want to point out that we use an hydraulic conductivity of the sand of 10 ala minus 4 meter per second, which is actually very high for sand. So it's a very conservative estimate of what it may happen. The same things we can do now for the resident time. Here is the mean resident times. Here is the standard deviation of the resident time distributions, and we can link then their distribution with the same information. So in this case is the ratio between the mean velocity hyporheic flow, which is linked with the morphology and the groundwater. So it's dimensionalized by it. The important information that we gain here is that once we have just a 0.2% of groundwater compared to the hyporheic flow, we have a very drastic reductions of the resident times into the stream bed. That means that the flow line are nicely packed toward the stream bed, and that impact also the distributions. What is interesting is that the median resident times, so 50% of the flow line have less than it is resident times over here, are less impacted by the groundwater effect. And that in a certain way is because the fact that the resident time distribution is mostly log normal with a very fat tails, and the median resident time usually is less impacted, or the median value usually is less impacted than the mean value by changing the tails of distributions. For a pool riffles, we can do the same things. We can build another model which can correlate the flow field and the morphology of the rivers. And in this type of simulation, we use the model developed by Colombini, Seminara, and Tubino, where given a full material that is loose and the bed form fully covered by the flow, we can predict what is the pressure distribution over the bed, and from that we can derive now analytical solutions of the flow of the mean downwelling flux into the sediment as a function of the relative submergence, which is the ratio between the hydraulic depth of the water and the amplitude of the bars, and here a dimensionless velocity, which is characterized by the mean velocity and then the Chézy, the dimensionless Chézy coefficient that tell us the roughness of the bed, and then the hydraulic conductivity. Notice here in these other graphs where I plotted the ratio between the hyporheic velocity here divided by the velocity. Notice the impact of ds. ds is the dimensionless grain of the particle, which is the ratio between the d50 and the depth of water. The rougher the system is, the more flow we go there. And it's not because of the change of the hydraulic conductivity of the sediment. It's just because of the configuration that the pool or riffle will take as the material gets coarser and coarser. For pool riffles, we can develop the analytical solution for the resident times, both in terms of the mean, medians, and the standard deviation, linked just with the information of the depth of the water and the and the amplitude of the riffles once the pooling riffles are in equilibrium with the flow. Here we have the dimensionless times that account for the hydraulic conductivity, the slope of the streams, the roughness through the Chassis coefficient, and the size of the pool riffles with L, which is the length between two consecutive pools. Do this type of analysis works, how well they do work. So we apply our biogeochemical model with measurement done by Bollier et al. In 2009 on the Kalamazoo rivers. They collected emissions, production of nitrous oxide, which is a very potent greenhouse gas, is 300 times more potent than CO2, and is a byproduct of the nitrification of nitrogens. The triangle, red triangle, is their measurement, and meanwhile the circle, the blue circles, are our model prediction. Basically, for each path line, we calculated the transformation, and then we integrate through all the size of the reach, the emissions of nitrous oxide for each upwelling areas. The model is not calibrated, so we just took the hydromorphological information that were measured when they did that measurement. So it's just the model. What about heat transfer? We did a set of experiments where we buried the Tibit, which is a temperature sensor, in the Bear Valley Creek in Idaho at 4 different depths. We had one into the streams. One was 15 centimeters below the stream bed, another one 25, and the last one at 50 centimeters. We use the one on the top to characterize the water, the temperature of the water surface, the one at the bottom to characterize the groundwater flow, such that we have a close boundary condition for the advection diffusion equations for the heat transfer, and then we we predicted the temperature at one point, at the basically there are 18 points at 2 depths, so the one at 15 and 25s, from just the information that we collected into the field. So we measured the water surface elevation, and that was the driver of the hyperec flow. And here is the index of agreement between at 2 different depths for what we predicted and what we measured. So we have now a set of tools that works very, very well. However, the question that we have is this one. This type of modeling works extremely well if we are working at the reach scale. However, what we would like to know is what happened at the network scale. What are the thermal regimes of the hyperec flow through all our watersheds? And what are the biogeochemical reactions happening at the watershed scales? If we are able now to connect the morphological characteristics of the surface water with the hyperec processes, now we can start to create a framework that uses the analytical solution that we just built at the reach scales, such that we can have that type of information. And to do that, we suggested a Damkohler number, which is the ratio of 2 timescales. The first timescale is the timescale of the median resident times of the hyperec into the flow, into the streambed, and that is a function just of the river morphology. So it requires only hydromorphological information. The second time scale is the one of the process that we are interested on. So it could be, what is the redox condition of the hyperec zone? Is it reducing or is it oxygenating? And in that case, we are going to use oxygen as our time limit. And it could be the times required for dissolved oxygen to reach a concentration below which the system is anoxic. Another one could be for thermal analysis, and we could take the times required for the daily amplitude of the temperature into the streams to be reduce that amplitude of 1/E. So we can define our value. If we do that one, and we use the one of dissolved oxygen, then we can divide at the global scales of the reach areas if the reach is mostly in aerobic or anaerobic condition, because a value of DI less than 1 means that most of the stream bed now is in aerobic conditions. A value that is larger than 1 tells us that the system is mostly in anaerobic conditions. And if it is in anaerobic condition, means that we have mostly denitrification into the streams. With this information now, we can create this type of graphs which are purely analytically, and depends on the river morphology. And here we are capturing the local characteristic of the streams. Here we are capturing both the hyporheic characteristics, so through the morphology, and the biogeochemistry of the systems. And we can see that the streams, so that will be in this area here, will be mostly in an aerobic condition. Meanwhile, over here, the hyporheic zone will be mostly in anaerobic conditions. This is constructed now for dune, and this one, sorry, for pool reef-fall system, and this one is for dune. Notice that I've been talking only on dunes and dunes and pool reef-falls, because they are the only two bed forms of which we do have a nice analytical and theoretical framework to study hyporheic flow. We can also take into account if the system is not neutral, so if we have upwelling and downwelling due to gaining and losing streams, so we can adjust for that. And we can also work for determining if the thermal property or the thermal regimes of the hyporheic zone depends on the daily fluctuation of the water temperature, or if not, because here, if the thermal Damköhler number is less than 1, means that in order to study the correct biogeochemistry reaction that happen in the streambed that are a function of temperature, I need to use the daily fluctuations. However, if my streams is over here, so it's larger than 1, I can use just the mean daily temperature in order to capture the main or the bulk response of the hyporheic zone. So to test our hypothesis, if we can create a theoretical framework to study at the regional scale, we use the LYNX-2 dataset. The LYNX dataset is a unique dataset because they measure N2O productions in streams in 72 different type of streams that goes between arid to tropical forest. They go from urban, agriculture, reference streams. They have through 9 biomes, so it gives us a lot of variability, and a strong model should be robust in all of them. The first things that we did was to normalize the flux, the production of N2O with the reactants, so at the amount of load that is there. And if we do that one, what we realize is this one: the impact of land use start to disappear, because the main contribution of the land use is actually on the flux. If we dimensionalize it and we do a statistical analysis, all three, they are not statistically different. And then if we then apply our theoretical framework, what we do have is that now the dimensional and the flux of N2O now scale linearly with the Damköhler number that is defined as the median residence times and the town limit for the dissolved oxygen. So now, only knowing geomorphology of the streams and the ecosystem respiration, we can determine how much production of N2O we have into the streams. And that is a very powerful tool to have at least at the first order what is happening into the streams. I wanted to show you the importance of morphology. Over here we have the two systems for which we have an hyporheic model, is the pool, reef-falls, and the dune systems. Both of them collapse exactly on the same universal law, which is this red line. However, the step, pool, and cascade, we don't have an hyporheic law for those ones. So what we did is to assume, because we know a step and we know the length between steps, let's use the pool reef-fall system for those ones. The pool reef-fall system captured basically the physics of the system, but not the scaling of the system, because it's not the correct closure for it. And because of that one, they all lie on the same straight line, but they do not collapse on the red one. We assume that if once we do have the correct hyporheic model for those two morphology, the blue one should collapse over the red one. What are these squares? These squares are rivers for which we have no information. We didn't know if they were plain bed, we didn't know if they were cascade or pool reef-fall. So we applied to them arbitrarily pool reef-fall systems. And because of that, we have a very large scatter. So morphology of the streams is very important in this type of systems. The other thing that I would like to show to you is about the heat transfer. We developed a very quick way to measure actually scour and deposition in streams using just the temperature as a signal. So if you have two sensors that measure temperature, one into the stream and one into the bed, and then we can calculate what is the change over time of the thickness of the sediment between two sensors once we capture the thermal property of the systems. This thermal property can be calculated in the first set of the experiment, and this one is become basically time dependent, from which then just from information of how the daily temperature distributions is changing over time, so we can characterize what is the erosion and deposition. And we tested that in Idaho with a set of control erosion and deposition. Here in green is erosion and deposition that was scoured manually, and the red and the black one was the one that we predicted just using temperature. And then we did the same analysis in below a dams in order to see scour and deposition below dams. And here we have the red, the black one is when it was measured at the beginning, the green one is at the end. Meanwhile, the dashed red line is what we predicted using our model, and we can see that at the very beginning and the end we match what was observed, such that we can now predict through an event, a flood event, how the scour and deposition processes occurs. And thank you for your time.
Thank you, Daniele. So actually, hang on. I'd like to invite both speakers back up for the question period. We'll open the floor to questions. And I also want to note that this session is a little different than the others. We have a third speaker later on this afternoon. And after that, we'll invite all the speakers back. So if you have more questions for Daniele or Dr. Takaman, we'll have time for that. Thanos. And if I could have each of the speakers speak into the microphone when you're answering questions. Is there a, let's down there for Thanos.
Thanos Papanikolaou, University of Tennessee, for Danielle. I really enjoy the presentation, actually both presentations, but Danielle, I like how you approach the ecological framework that you are developing, or using basically hydrogeomorphology, and then bringing into picture the biochemistry. My question to you is, have you performed any measurements for various relative submergences? And looking the dam color, number, and other parameters for the various relative submergence conditions.
Yeah, I think that is possible, and that depends on two things. So far, all the analysis that we present are based on the concept that the bed forms is in equilibrium with the flow. So we have also the dunes that moves or they don't move, but their size is scaled with the hydraulics property of the flow. We can do the same type of analysis where once and what happened also in streams, once the dune or the bed form is forms, and then we have a lowering of the flow, sometimes those forms get frozen. And so at that point, we could develop another type of set that characterizes and links at that point the frozen bed forms with the surface hydraulics. Yeah.
Bill.
Bill Dietrich, Berkeley. Really enjoyed both talks, but I want to ask Dr. Takaman a question. The association of life with habitat is, what you've done is very impressive in defining the conditions favorable for a particular organism. I'm wondering whether you have, and just didn't show, or considered taking this further. The experience in the Pacific Northwest on salmon suggests that you really need to look at the entire life cycle and then connect the life cycle to a population dynamics model. An example is that we tended to focus on salmon spawning grounds. When we started doing watershed analyses and geomorphologists went out and would characterize where the salmon spawning grounds and worry about industrial forestry releasing sediment and degrading spawning habitat. But then when studies were done to look at the full life cycle, it turns out the salmon are prolific. They produce a lot of eggs from a few spawning sites. And what turned out to be, if you follow the individual life cycle, is that overwintering habitat may be limited. And the only way to discover that is both through monitoring the organisms through their life cycle and then modeling that to find where the limiting factor was. So, and that approach could be applied to the mayflies, the gobies, the ayu, all of the things you've illustrated. So I'm wondering whether you've considered that, and because at least the experience I've observed in Pacific Northwest is that not simple, but the association of habitat and organism is incomplete and could lead to a decision on management about focusing on the wrong thing. That you need, there may be some limiting factor in the life cycle that you haven't seen that even though the spawning habitat may be poor, that's not the limiting factor. And the other part of that is that, and even more difficult, that's probably to bring in is food webs. That is, it's not just that you have to exist, you have to eat, and you need to, find out where your food is, and an organism may be limited by food availability rather than association. So have you pursued that kind of approach?
Okay. It's a very tough question, but I should answer a bit. For the first point of view, of course, each organism has their life history, and each stage they need different conditions of the habitats. It means, for example, as to the salmon, the reproductive stage has the limited by the spawning site. I think from the aspects of the management, this is one of the potential, and this potential should be high. Of course, the other aspects also should be considered. But anyway, for the purpose of the reproduction, this stage have to, this kind of properties should be considered. And such kind of way we can make several requirements for each stage. And of course, the desirable conditions should be considered all. However, for each stage, we should consider these characteristics, and we should consider these characteristics. Then, for the management, we can say that image of the consideration. We can say to the manager or the citizens, this is one of the solutions for your questions. And another one, of course, the ecosystem should be conditions should be considered as very many dwellers, and that's why only one species is not enough for consideration. Of course, but for example, as to the eyefish I focused on, this is anadromous species, and this is one of the many anadromous species, and it means that if they can survive, other anadromous species also has the benefit from the management. So, of course, we cannot consider all of them at once, at one time. But if we select the several kinds of the species required with the different aspects, then as a total we can get the better biodiversity. So for this purpose, we have to make the monitoring of the biodiversity, and we should select the proper species for the target. But if we think about the whole, there is not always the good solution for the management, because we cannot have the good information for the concrete measure for growing up in the high biodiversity. That's why I selected this particular species, and the set of the particular species must be the solution for all. Yeah, this is my opinion. Thank you for your question.
Marco Redolfi from the University of Trento. I have a question for Daniele. Is it usually possible to assume in a gravel bed river that the residence time is much shorter than the morphological time scale? Or are the two time scales comparable so that you cannot assume a steady flow, a steady morphology for your solutions?
That's a very, very good question, actually. From a set of experiments that we have done in Trento and also a numerical set of solutions that we did in IDO, what we noticed is this one, that once the flow that form the bars then slightly subside and the bars start to migrate, but very, very slowly. Now the resident times that happen into the bars start to be smaller compared to the migrations of the bar itself. In the case instead that we are keeping the same form in discharge, then the concept will break down, because at that point the scale of migration is actually faster than the hyperect time scale. Yeah. Thanks for asking, actually. That's very important. Yeah.
Colin Thorne, University of Nottingham. I was particularly struck by your diagram, Yoshiro, showing the downward control of the flow and sediment regimes on geomorphology and habitats, but also the upward control of ecosystems and species on geomorphology and habitat. And you didn't talk much about that.
Yes, yes.
A couple of examples that I do know quite a lot about are crayfish and caddisfly larvae, both of whom work actively on the benthos to improve it for their life cycle and their own purposes. The crayfish, the signal crayfish particularly, are very territorial. They build castles, they build fighting pits, and they disturb the surface armor and work at Loughborough by Steve Rice and my colleague Matthew Johnson suggests they can lower the critical shields parameter by 40%. Conversely, caddisfly larvae glue the bed together using underwater silk, stronger than tensile steel for catching plankton. They can they can increase the shields parameter by 40%. Is that significant? I think probably it is as a geomorphologist. And my understanding, and I don't know enough about this, is that salmon also preferentially pick spawning sites with strong positive hyperoic flow because it's better for their eggs. And they're not conscient beings, they don't know this, but it's an evolved behavior. It's something they've learned to do over millennia, millions of years. So of course they're very good at it. Could you both perhaps comment then on the role of plants and animals at the bed, not as dependents of geomorphology, but as living beings that influence geomorphology, and I think probably very significantly?
I think there are several methods for the organisms to make the manipulation of the environmental conditions, and the caddisfly is the one of the very famous one, and but the another one may be the migrant or the dwellers inside the sandy bottom. If they make the holes, and if the frequency of the movement of the sand is very frequent, then the water contents and the permeability or the space of the pore should be more after creation of their nesting behaviors, and that's why this part becomes very erosive easily. And such kind of conditions usually occur in the lower stream of the rivers, and some tube builders sometimes has the different another impacts. The tube builder making a lining of the tube, then the tube is very stuck and it is very difficult to erode. And if this kind of species are abundant, the sandy flat cannot move easily. And such kind of different directions will be occur dependently to the dwellers. And you see the caddisfly will become the latter, and mayflies become the former. Yeah.
Yeah, on the Salmonis part, we just got Marijuana and Todd Baxton a publication looking at the impact that Salmoni had on the mobility of particles. And we noticed that the mass spawning of small-bodied Salmonis, actually what they do is to enhance the mobility of the stream bed. primary one. So in the case of low slope with fine materials, is mostly the dunes, and those one interact directly with the benthic and the hyporheic zones. In gravel bed river, we take account partially of the plan where we are analyzing the system as a 3D, so as a three-dimensional flow that goes through the riffles, so to move underneath a bar, and then due to the change in hydraulics, we move to the other one. So we have that 3D. What we do not account for is the hyporheic flow that developed between meanders, and the reason is because exactly what you pointed out, that the time that takes for the water to flow through the meander is very long compared to the time scale of the biogeochemical reaction that we are looking at. Yeah.
But others have done that calculation.
Yes, there is a work done by Cardenas on the amount of nitrous oxide that can be formed between meanders.
Okay, I'd like to thank the speakers once again.